Once you move on past the sponges, you end up, the next group that branches off of our little phylogeny is a group called the Cnidaria. And really there are a couple of other phyla around here, kind of splitting off the tree at about the same place, but they are relatively minor. The Cnidaria are not. They're actually a pretty big, pretty important group. And there are some very interesting evolutionary steps that were taken between this ancestor here and this one here, because the Cnidaria have a lot of features that sponges do not have. There's a theme that I want to introduce at this point that, again, is going to pop up multiple times as we talk about animals. It is the lesson of the spork. You may be familiar with sporks. I think of them as the utensil of betrayal, because a spork promises that it is going to be really handy. Hey, you could use it as a fork or you could use it as a spoon. But whenever we were given sporks with our lunch in high school, I found that it'd be deeply disappointing, indeed frustrating, because if you try to stab food with a spork, like you would with a fork, you actually really can't do it. It's very difficult to do. And if you try to eat soup with a spork, the way you would with a spoon, you can't get very much soup in it because those little tines, the openings between the tines, just let all the soup run out. So basically what a spork is supposed to be, it's supposed to be two things at once, but actually what it ends up being is nothing at all, and not being good at either one of those tasks. Very often, a compromise between two different designs ends up being able to do neither task well. And that's a lesson that we're going to be seeing. There are solutions to this, which life has been very clever in coming up with. The phylum Cnidaria includes a lot of animals that you're probably familiar with. The sea jellies are only one of those. There are also anemones and corals are other animals in this group. The Cnidaria are part of a group of animals called radiates. And they have this name because their symmetry is radial symmetry. Again, if you look down on top of a sea anemone, you won't find just one plane of symmetry. You can find an infinite number of planes of symmetry. They do not have a lot of the organ systems that we associate with animals. There's no respiratory system, no circulatory system, no excretory organs. Having said that, though, they are much more organized in body form than a poriferan is. Just as the collar cell is a type of cell that's only seen in the porifera, the cnidocyte or cnidoblast is a cell type that is only seen in the Cnidaria. This is the cell that gives this phylum its name. Cnida is from the Greek word for nettle, as in stinging nettle. What the cnidocyte is a cell that contains, it's a cell that contains an organelle called the nematocyst. And these can have different functions, but the basic, or they can have different forms, but the basic form is this long inverted thread curled up inside this organelle. It is basically a cross between a landmine and a poisonous harpoon gun. What happens is that when a nematocyst is triggered, there's an extremely concentrated solution inside this organelle. And when triggering happens, water is allowed to rush in, increasing the pressure very dramatically inside that organelle. This thread is actually inside out. It's like the inside out finger of a glove, as though you were to take a finger of a glove and push it down into the body of the glove. When the pressure builds up suddenly inside the glove, that finger turns very rapidly inside out, or right side out rather. The acceleration, I mean, it's a small piece of matter, but it can accelerate up to 5 million times the acceleration of gravity. So here's what it looks like. The top to this little weapon opens up, water starts rushing in, and that starts turning this tube right side out. If you are unlucky enough to be an organism that just brushed up against this thing, that means that this tube is basically shooting out into your body. It's shot out with such force that it can penetrate your flesh. And all of this stuff, these little dots here, this is all poison that was on the inside of that organelle. Um, Cnidocysts or nematocysts can deliver an incredibly toxic sting, enough so that some of the organisms in this group are lethal to human beings. Obviously, one of these is not going to do very much because this is a microscopic weapon. But if you get stung by, say, hundreds or thousands of millions of these at once, it can be very damaging and indeed fatal. Accordingly, these cells are usually arranged in batteries, and that's what these little lumps are on the tentacles of these sea anemones. Those are batteries of cnidocytes. They all respond at once, so that just one trigger of that battery will cause them all to fire. And that can take out a much larger prey item than if they all had to fire individually. For the most part, they stab into you and poison you, although there are some other clever ways that these things can work. They can just stick to you so that you're glued to the sea anemone, and then it eats you. But usually it just poisons you and kills you and eats you afterwards. Some of these poisons are very nasty. Some of them attack the nervous system. And because of the way that the nematocyst is formed, it just sort of automatically injects them into your flesh. It's really quite clever if you're into that sort of thing. The cnidaria are interesting because unlike the sponges, they actually have true tissue layers. But they do not have all of the tissue layers that are in our bodies. The organisms that we're most used to thinking of, like ourselves, have three tissue layers. There's an ectoderm, an endoderm, and a mesoderm. The cnidaria never form a mesoderm. What happens is that in early development, the gastrula forms here, this hollow ball of cells with one opening into the inside. And that becomes two tissues. There's endoderm on the inside, there's ectoderm on the outside, and there's stuff in between, but it's not a tissue. Okay, the inner layer, the endoderm, develops into what's called the gastrodermis. The layer on the outside develops into what's called the epidermis. In between is glop. It's referred to technically as an extracellular matrix. It's called mesoglia, and basically what it is it's the jelly in a sea jelly. There are very few living cells actually in the mesoglia. It's non-living stuff that takes up the space between the gastrodermis and the epidermis. Why is this useful? Well, it can be used as a sort of skeleton. Like most fluids, mesoglia is not easily compressed. And so when you push against it, it essentially pushes back. It's bendy, it's flexible, it's squashy, but a sea jelly can use that basically to move its body around. It can push off of that jelly because the jelly is incompressible. This allows the jellies to actually swim. There are a lot of these animals that are sessile, at least during part of their life cycle, but some of them are capable of swimming around, with a certain amount of direction, although they're really quite hopeless compared to what you would expect in the swimming of, say, a vertebrate or an arthropod. The nervous system in these animals is very interesting. It's not centralized anywhere. It is a diffuse nerve net that is spread over the entire body. In some places around, in some of these organisms, it may form kind of nerve rings, but there's anything like what you would call a brain. And signals can actually be sent both directions along any of these nerve cells, so that there is no real way of gathering information all in one place and integrating it. There are two basic body forms within the phylum Cnidaria. And what's interesting is that a lot of cnidarians, not all, but a lot of them have a life cycle where sometimes the animal is one form and sometimes it's a different form. And these life cycles can be pretty complex. If you imagine a sea anemone, okay, that of course has its mouth and tentacles at the top, that's a polyp. If you imagine a sea jelly that has the mouth on the underside and the tentacles hanging down, that's what's called a medusa form. So some of you may be familiar with the animal Hydra. Hydra doesn't form medusas, it's just the polyp form. It's kind of your classic cnidarian polyp. Here's a very close-up shot of one of those. This is also a polyp. Sea anemones you only see them in the polyp stage. Corals also are polyps, but in this case they're colonial. So each one of these tiny little, they're almost like tiny little sea anemones, is a polyp animal, but they're all genetically identical and joined physically together. This is the structure of a polyp. You've got the single opening to the gastrovascular cavity, which is lined with gastrodermis. Sorry. On the outside is the epidermis, and there's a thin layer of mesoglia in the middle. Polyps can reproduce in a couple of different ways. They can split in half, or they can do what's called budding, which is shown here. This is a little baby polyp that is just stuck onto the side of its parent. It's just been kind of growing there. Here you see another bud that is about to become a third polyp. What can happen in organisms like corals is that these polyps will grow, but they don't actually physically separate. They remain connected to one another. In that case, what you have is a cnidarian colony. Interestingly, when you have multiple polyps connected together, they can take on different forms and different roles. This is a phenomenon called polymorphism. It can be extremely useful. So for example, this is a colonial cnidarian, and these zooids, these different polyps, have taken on very different forms because they do different things. Over here you have a gastrozoid, a feeding polyp. It has the stinging tentacles all around it, and the gastrovascular cavity where it can take in and absorb the food. Over here you have a gonozoid. This is a reproductive polyp. No tentacles. Instead, it puts all of its energy and resources into reproduction. So you've got specialization within the colony. And this is one of the ways that an organism avoids the problem of the spork. Instead of trying to do two things at once and doing them both badly, if you have a colonial form, you can designate some of the individuals within the colony to be forks, and other individuals can be spoons. And the forks are really good at being forks. They actually work like good forks. The spoons act like good spoons. You can pick up soup with that spoon, right? You don't have to have this in-between spork that is good at neither one. All you have to have is a bunch of genetically identical individuals that work together as a single organism. Okay, so you can do without the spork if you have different individuals within a colony specializing on different jobs. The medusa form is a different body form than the polyp, but basically it's built along the same lines. Here the mouth is upward, here it's downward, but if you flip over a medusa, you can see basically the same structure. Mouth into the gastrovascular cavity. The gastrovascular cavity lines the body here. There's epidermis on the outside, mesoglia in between. The only difference is that this polyp is kind of pulled downward into this stalk or column, whereas the medusa just has this big rounded blob of jelly on top. Medusae are different than polyps. Most polyps do not move around. They're what's called sessile. Medusae are the things that you call sea jellies, or some might call them jellyfish, except that that's a really dumb name because, seriously, jellyfish are not fish. They're sea jellies. The medusae are the things that you see swimming around. They're not great swimmers. They're not coordinated. They do have some sensory ability. They can sense light and dark, so for example. And so they do tend to swim a little bit directionally, but their abilities are pretty limited. This is the life cycle of a cnidarian, or at least of some kinds of cnidarians. And the reason I want to show you this is because there's a second way of avoiding the problem of the spork. Let me just talk a little bit about how the life cycle works. In these cnidarians, you first start out with a larva. This is a tiny, tiny microscopic thing. You can see the individual cells on it. But this larva finds a place to settle, and it metamorphoses into a different shape. It becomes a polyp. Starts out as a blob, eventually forms what's called this primary polyp here. Okay, and that looks more or less like a hydra, this animal that you saw earlier. The role of this thing is to sit in one place. Hopefully it's found a good place to live and grow, and feed and grow and feed and grow. This is the food collecting stage during the life cycle of most cnidarians, although not all. That polyp may bud to form a colony, and that colony may have different zooids, different polyps, doing different things. But so for example, again, here's the gastrozoa that collects food. This is a gonozoid that buds off reproductive structures. And in this case, the reproductive structures are medusae. So this colonial form, this colony of polyps, is making tiny, tiny little sea jellies that float off into the water column. What the role of those is to spread far and wide and make sperm and eggs. This is the sexually reproducing stage in the life cycle. Once they form the sperm and eggs and those sperm and eggs meet, you end up with egg cells that develop into the larva, which then starts the cycle over again. What does this have to do with the spork? Well, what you're seeing here is not only specialization among individuals within a colony, but specialization in time as you go through the life cycle. This form here is specialized for sitting in one place and feeding. It is capable of feeding and growing, getting a hold of the nutrients that are necessary to build a large reproductive colony. But this life form is different. It has a different shape. It can swim. And its job is to travel long distances and find mates that may be some a long way away. If this polyp had to do that job, it would be very poor at traveling around. If this medusa had to do the job of feeding and growing into a colony, it wouldn't be able to do that because it wouldn't be able to swim while budding off all these other different individuals. So the genius of this life cycle is that you've got these necessary parts of organismal function divided among different forms, different actual individuals within the life cycle. All organisms have to feed and grow and reproduce, but you avoid the problem of the spork by sort of fobbing those jobs off on different organisms that are adapted to those specific tasks.